Hello children, let us continue our discussion on electron transport chain. Today we will be talking about complex 3 and 4. In the complex 1 and 2, as we have learned, a total of 4 electrons are passed to ubiquinone, reducing it to UQH2. To run the Q cycle, which involves oxidation and reduction of ubiquinone, the reduced form UQH2 must be oxidized back to UQ. This is important because if oxidation of UQH2 does not happen, the electrons from complex 1 and complex 2 of ETC cannot be passed on, leading to either slowing down or the halt of Krebs cycle. This is because NADH will not be participating in transfer of electrons. Moreover, succinate will not be able to convert to fumarate. Therefore, reduction and oxidation of ubiquinone through Q cycle is important. Complex 3 is involved in this cycle. Complex 3 does the job of removing electrons from the reduced form UQH2 and transfer it to cytochrome C. Cytochrome C is a small mobile electron carrier present on the positive side of the inner mitochondrial membrane. It shuttles electrons from complex 3 to complex 4. It thus connects the two complexes of the electron transport chain. Coming to the structure of complex 3, it has a heme moiety of two types, B56 and B560, along with cytochrome C1 and FES proteins. The complex has two binding sites for ubiquinone, QN for the negative side of the membrane, which faces the matrix, and QP, the positive side, which faces the intermembrane space. The complex operates in a manner where two electrons are removed from UQH2. One of these electrons is passed to cytochrome C1 via FES, and then the electron is given to cytochrome C. We will soon learn that this electron is then transferred to complex 4. These reactions happen at the QP site of complex 3. The second electron at QN site is received by ubiquinone via cytochrome B1 and cytochrome B2, and ubiquinone now becomes semiquinone, UQ with one electron. This cycle is repeated twice, and the semiquinone then becomes UQ with two electrons, and it is a partially reduced form. This molecule then picks up two protons from the matrix and is finally reduced to UQH2. The reduced ubiquinone is freely mobile in the membrane and gets oxidized at QP side after giving 4 protons per electron pair to the intermembrane space. This is Q cycle, which is responsible for proton transport across the membrane. Let us bear in mind that for the transport of a pair of electrons downhill, 4 protons are pumped into the intermembrane space by complex 3. Next is complex 4, the cytochrome oxidase. Children, till now we have come to the conclusion that NADH, FADH2 are produced and they are being processed through electron transport chain. We've already done Krebs cycle, glycolysis, link reaction. But if we talk about aerobic respiration, we have so far not learned where actually the oxygen is involved. Though aerobic respiration has to have the consumption of oxygen. It is this complex 4, that we will learn is involved in the entry of oxygen, and we will do its functions here. Complex 4 is the site of oxygen reduction. It is here that oxygen accepts electrons and gets reduced to water. The following is an account of the path of the electrons which lead to reduction of oxygen. Cytochrome C, which has accepted electrons from complex 3 and is a mobile carrier, is present on the intermembrane space side. The redox centers for the complex 4 are cytochrome A and A3. There are two copper centers, copper A and copper B. The electrons from cytochrome C1 will move towards the complex 4. The path is given as from cytochrome C, they move to copper A, then to cytochrome A. From there, they move to copper B, and finally to cytochrome A3. From cytochrome A3, the electrons are given to oxygen, which gets reduced to form water. In the process, two protons from the matrix are pushed into the intermembrane space. Hence, complex 4 is involved in pushing two protons to the intermembrane space, reduction of oxygen to form water. This is a simple diagram to show you the accumulation of 10 protons on the intermembrane space. 4 protons, if you remember, are given by complex 1 when NADH gets oxidized to NAD. 4 protons are given by complex 3 when the Q cycle runs, where a pair of electrons requires 4 protons. 2 protons are given by complex 4 when a water molecule is formed. There is no proton pump activity by the complex 2. So this is how the proton gradient is built up across the inner mitochondrial membrane. Next, we will be doing how this proton gradient is utilized to generate ATP finally through the ATPase pump.